Thanks for joining us. Today I'm going to tell you about near perfect predictions of horizontal gene transfer and the emergence of antibiotic resistance. So bacteria harbor plasmids, and they're most notable because they carry lots of antibiotic resistance determinants, but they can also carry other types of genes like mercury resistance genes and metabolic genes that allow them to metabolize specific substrates and survive in different types of environments. So we can learn about bacterial ecology by studying this process. Within bacterial communities, the movement of DNA happens through conjugation of plasmids through pili, the uptake of DNA from the environment through transformation, and through phage transduction through viruses. So in order to study these bacterial communities and plasmids, we'd like to know how these bacteria, how these mobile genetic elements, including plasmids, are getting around between bacteria. So the question that I'll tell you about today is this HGT network predictable? Other people have proposed several hypotheses. First, that organisms which are similar can share DNA. They have to be within the same ecological niche. They also have to have a certain amount of machinery that's compatible. Their genomes have to be compatible, and they have to not have these defense mechanisms that restrict the movement of DNA. So people have tested each one of these things individually, but in my lab, we had the idea that if we used the gene content of organisms, we could potentially integrate across all of these different hypotheses. To do that, we created a network of bacteria. We tried to create a very diverse network that wasn't biased by reference databases. This contains over 10,000 species and over 12,000 genomes sampled across the tree of life. And each of these links represents a gene transfer event. So the presence of identical DNA in distantly related organisms. That's the proxy that we used, which has been previously vetted. So we performed a logistic regression using phylogenetic distance, 16S distance, and with that, you get pretty good predictions of the linkages within this HGT network. But when we use KEGG orthologous groups, which are protein families with specific functions, we get very good predictions, even better using a lasso, and then really good predictions using a random forest machine learning approach. We tried doing this with ecological distribution and 16S distance, but we didn't get as good predictions as when we used just the functional content of these organisms. We were able to look at the important features, and we do indeed see that the gene content integrates across these different hypotheses, and that specific genes wind up being important. So we have antibiotic resistance genes that are important for predicting transfer, as well as certain niche-specific genes, things like anaerobic enzymes, which indicate organisms that live in either an anaerobic or aerobic environment, and other types of metabolic genes in some cases. Yeah, you can see the list here. So things like heme biosynthesis, purine metabolism, sulfur relay display, probably also a niche specific enzyme. Okay, so the next thing that we tried to do was apply a different type of machine learning approach, a graph convolutional neural network to this problem. We get very, very high AUC values, really good predictions. But the place where this graphical convolutional neural network works is that it improves the prediction of between phyla transfers of DNA. Okay, so linkages in the network, HGT linkages between phyla, these are better predicted with this GCN model than with a random forest model. And this GCN model, we're increasingly giving the model kind of a peaks at more and more links in the network. The reason that this is important for microbial ecology is that these long distance transfers between, here is two depicted between Proteobacteria and firmicutes and Proteobacteria and Bacteroides. These are where you get these long distance transfers, which are relatively rare within the network, because I already told you that phylogenetic similarity is important. They're relatively rare, but once you have one of these events, you have a new gene function that's now present within a nucleate where it's more easily transmitted between bacteria. So we think that these are important for the evolution of these organisms and the emergence of new traits, including antibiotic resistance. When we predict antibiotic resistance, gene transfer events specifically, our predictions get even better. So we decided to predict, see if we could predict which specific antibiotic resistance gene, according to antibiotic class, was being transferred. And you can see these different genes are predictive of the different classes. When we look at which genes they are indicative of the mechanisms of these different antibiotics. So for example, glycopeptides, which target gram-positives, we can find spore-forming gene or sporulation genes that are important in predicting those linkages, which makes sense. Similarly, we see genes involved in membranes and cell walls being important for the prediction of beta-lactams, beta-lactam resistance genes. And beta-lactams are important for cell wall, or they inhibit cell wall production. And so that makes sense as well. We also see lots of genes involved in the mechanisms and the machinery of gene transfer, and also the inhibition of gene transfer, things like CRISPR-Cas and toxin antitoxin systems. And we think that when we're looking at antibiotic resistance genes specifically, these kind of restrict or kind of they're indicative of the mobile element itself that these genes are being transferred on, and they're probably more restrictive and represent specific subsets within the network. Next, we tried to ask whether if you're some researcher studying a specific environment like the ocean, soil, plant, or human microbiomes, and you have a bunch of genomes that have been selected, could you use our model built on many, many organisms to predict gene transfer within your specific community? And even though these microbial communities have fewer species and also fewer of these functional groups when compared with our original model, in every case, when we predict by 16S compared to when we predict by random forest using these functional groups, are in every case, our predictions improve. Again, validating our method. So I hope that I've convinced you that using machine learning approaches, we can predict the HTT network with high accuracy. Anyway, we're recruiting my lab, so if you liked this, please look us up. Here's our website. And I just want to thank the people in my lab, specifically Hao Zhu, who did the work, and then all of our sponsors. Thank you very much.